Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. How are you, dear students? I welcome you in Pakistan International School, Taif, and this is virtual learning class for subject biology for first year students. Dear students, today is lecture number 38 and we are gonna start our next chapter, that is prokaryotes. This is chapter number 6 of your book, and in this chapter, we will talk about the primitive organism that are prokaryotes. And we know that what are prokaryotes means the organisms which do not have proper nucleus in their body. They are unicellular organisms, and we will talk about all these organisms in detail. And today's topic is taxonomy of prokaryotes, and then we will see two main categories of prokaryotes, that is archaea and then bacteria. Dear students, prokaryotes, that is a unicellular organism. They lack an enveloped enclosed nucleus. So it means there is no nuclear membrane, no proper nucleus is there. The word prokaryote comes from the Greek word pro, it means before, and karyon means nut or kernel, means around their nucleus there is no envelope, no membrane, no nut like structure, and these organisms just come and arise before the eukaryotes, which are having the proper form of the nucleus. Taxonomy of prokaryotes, that is like lots of scientists have done their work on that. So the first one, the Dutch scientist Antony van Leeuwenhoek, he discovered bacteria first in 1674. He used the single lens microscope, which was designed at that time by his own, and he called those organisms which he found at the time animalcules. Ehrenberg then introduced the name bacterium in 1882, like 2 centuries after that he gave the name bacterium. And actually this name bacterium is derived from the Greek word that is called bacterion. It means that small stuff. So later on, the name is changed into bacteria. So bacteria is a plural, or bacterium, or we say, or bacteria, we say to that organism which do not have proper nucleus in their body. There are two separate systems of classification which are followed, in which prokaryotes are accommodated in different ways. So if we just see their taxonomic position or the classification, so they are classified as kingdom Monera, and then domain bacteria. And like in one classification is kingdom classification, the other is domain classification. What is that, we will see in this topic. So, in traditional two kingdom system of classification, all microorganisms, including prokaryotes, were placed in plant kingdom at the time. So, Sir John Hogg proposed the kingdom Protista to accommodate all these microorganisms. So, at that time, the first of all, kingdom prokaryotes were placed in kingdom Protista all together, in which viruses all unicellular eukaryotes and all prokaryotes, just they were placed under Protista. In 1866, Ernest Haeckel he proposed another group for those kind of organisms which were having slight difference in their in their cells. So, like he proposed three kingdom system in which the kingdom Monera was defined for the organisms prokaryotes. So Herbert Copeland elevated the status of Monera to kingdom level. So before that was not kingdom, that was just a separate group. But later on that was derived as a proper kingdom Monera. Robert Whittaker that he proposed when he proposed the five kingdom system, he incorporated the basic prokaryotic eukaryotic distinction. So he proposed the kingdom Monera for prokaryote as a separate kingdom, and it was defined and modified by Lynn Margulis and Schwartz in 1988. So they also designed a separate kingdom Monera for all prokaryotes. So it means lots of scientists are in the favor of separate kingdom for animals which are included in prokaryotes. These are not animals that we can say that these are unicellular organisms which lack proper nucleus. So the other one, we can see the taxonomic position of prokaryote as domain bacteria and domain archaea. So the term bacteria traditionally applied to microscopic single-celled prokaryotes. So molecular systematic studies showed that prokaryotic life to consist of two separate domains. So these domains are that is bacteria and archaea. And there is another domain because these two are having prokaryotic organization. The next one is eukarya, which is having eukaryote organisms which are having proper nucleus in their cell. So these three domains are now systematically studied under the classification or taxonomy. Now we will see phylogenetic position of prokaryotes. So evolutionary relatedness among various group of organisms is called phylogeny. So a major step for this study in 1977 that archaea has a separate line of evolutionary descent from bacteria. So that evolutionary or that we can say the phylogenetic taxonomy is based on encoding of ribosomal RNA, and this ribosomal RNA recommended as molecular clocks and divided prokaryotes into two evolutionary domains and three domain system like eubacteria, archaebacteria, and eukaryotes. So in that, gene sequences can be used to reconstruct the bacterial phylogeny, and the studies indicate that bacteria diverged first from archaea eukaryotic lineage. So now we will see its phylogeny in the diagram, then you can understand it properly. But here is the main point that phylogenetic position of prokaryotes are based based on genes encoding genes encoding ribosomal RNA. So that is called molecular clock. So, where the sequences of genes and nucleotides are gonna change from that area, the next group is going to be formed. So, phylogenetic position of prokaryotes. So in this system, two empire system arising from the work of Caton. Prokaryotes were classified within the empire of Prokaryota, but three-domain system based upon molecular analysis. As we have talked before, molecular analysis, or that is called molecular clocks, for constructing the phylogenies of these organisms. So, two domains like bacteria, that was formerly called eubacteria, and archaea, they are also said to be archaebacteria. Organisms with nuclei placed in the third domain, that is eukarya. So, in the study of origins of life, prokaryotes thought to be arisen before eukaryotes. So, here we can see in the diagram there are three domains: bacteria, archaea, and eukarya. So, archaea is the link between eukarya and bacteria. So here we can see the common ancestor, that is also called Luca. Luca means last universal common ancestor. Here is the common ancestor. So the scientists um, don't know about the proper form of that organism, but with this structure of tree-like structure, phylogenetic tree, we can. So in this phylogenetic tree, we can see that there are three domains. So bacteria are, we can say, the organisms, the very first prokaryotic organisms, which are actually the bases. And from here, uh, they are divergent, like common ancestor is divergent into bacteria and archaebacteria into two branches. So archaebacteria is closely resembled with bacteria, but it is closely resembled with archaea as well. And from here, the eukaryotes organisms arise from archaea bacteria, and here is also the common ancestor for archaea and eukarya. So eukarya, we know there are animals, fungi, plants, flagellates, ciliates, or protozoans are there. So in archaea and bacteria, there are different type of prokaryotic organisms. Now we will talk about archaea. Archaea are also called archaeobacteria, and you can see the structure. Any of the large group of primitive bacteria having unusual cell walls, membrane lipids, ribosomes, RNA sequences, and having the ability to produce methane. Proper eubacteria, they don't produce methane, but archaea produce methane, and they live anaerobically extreme conditions as well. So if we see the unifying features of archaea, so we can see here, first of all, cell wall. Very important, cell wall. The cell walls of archaea do not contain peptidoglycan, as we know that bacterial cell wall contains peptidoglycan, but they are different from bacteria by having don't have peptidoglycan. Some bacteria's cell wall is composed of polysaccharides like carbohydrates, and in others there is complete pure proteins, and a few of them they don't have cell wall. If we see composition of cell membranes, so plasma membranes of archaea contain unusual lipids that allow them to function at a high temperature, not as lipid bilayer, but their lipid is having a a different structure as compared to bacteria and other organisms. Like lipids of archaea contain glycerol linked with branched long chain of hydrocarbons, but as we know that lipids of bacteria they contain glycerol linked with fatty acids. The units of lipid is glycerol and fatty acid, but here in archaea the cell membrane is formed by glycerol and the branched chain of hydrocarbons. So that distinguish the archaea from other bacteria, as well as that just helps the archaea to live under the extreme conditions. Methogenesis, the ability to form methane in one type of metabolism that is performed only by some archaea. Mode of nutrition, that they are autotrophs, but they don't do photosynthesis. So they are not photosynthetic archaea. So what they do? They use molecular hydrogen, reduced elemental sulfur, carbon dioxide, and water. In normal photosynthesis, plants and some green bacteria they use carbon dioxide. We know carbon dioxide, water in the presence of light, they and chlorophyll they do the photosynthesis and produce glucose. But here they also use hydrogen and elemental sulfur for production of their nutrition or food. Ribosomal RNA sequence is the molecular clock of the archaea which distinguishes this from bacteria. So, the most fundamental and important difference between eubacteria or simple bacteria or archaebacteria is the nucleic acid. Which nucleic acid? Ribonucleic acid, and in that RNA, as we have said it, just molecular clocks. When the scientists have searched out their RNA, ribosomal RNA, so they found that in eubacteria, the sequence in at the place of 10th nucleotide out of 1500s. So there is the sequence UC, but in uracil and cytosine, but in archaea that is uracil and uracil in place of cytosine. So from here they got the difference, and that makes them another organism. So here we can see some differences between bacteria and archaea and ribosomal RNA sequence. RNA polymerase, so relatively small and simple. So that is in archaea that is complex, similar to eukaryotic. RNA polymerase, which the eukaryotic cell is having, they are having the same similar complex uh, ribosomal RNA. Sorry, RNA polymerase. That is an enzyme which make the RNA. Introns, non-coding parts of genes. So, for example, if okay, I'll just explain it. That is present in archaea and also in eukaryotic cells, but it is absent in bacteria. So we can see here, for example, this is DNA. When the RNA is formed from DNA, so what happened? There is, at some distance, there is useless genes. For example, this one and this one and this one. So that is called interons. Interon. So this interon is removed when RNA moved outside of the cell for messenger RNA, ribosomal RNA for production of the proteins. So it is going to be removed out and then it will form into the useful genes. So that is not present in bacteria. Introns are not present, but in that archaea they are present. They are also present in eukaryotic cells. So if we talk about the feature, antibiotic sensitivity to streptomycins and chloramphenicol, so that is like bacteria inhibited by this by but archaea doesn't. Peptidoglycan in cell wall in bacteria that is present but in archaea it is absent, and membrane lipids, that carbon chain unbranched is there but carbon chain branched in there in archaea. So some specific features for archaea is, number one, they are methogens, like they produce methanes as a metabolic byproduct in anoxic condition, like in the absence of oxygen. So they are strictly anaerobic organisms, are killed when exposed to oxygen, so they reduce carbon dioxide using um, hydrogen and release methane in swamps and marshes, so they can live in marshes in extreme conditions also. So they occur in the mud at the bottom of the lakes, swamps, etc. They're also found in the gut of some herbivores, cows, humans also. They present in dead and decaying matter, and it is very very important because they are added to biogas reactors for production of methane gas for cooking, sewage treatment, like for production of biogas. So its example is Methanophores, Equomeres, Methanophores ethanolicus, and Methanophores formosanus, and Methanophores liminetens. So they produce methane from hydrogen gas and carbon dioxide coupled to the formation of ATP, and also methane is formed, and that is utilized for the production of biogas. Then we'll see halophils. Halophils, halo means salt, phils mean loving or lover. So they require salty environment for survival. So they found in all salts lakes area with evaporation of seawater occurrence such as Great Salt Lake in USA and Dead Sea also. They can live in water with salt concentration exceeding more than 15%, and oceans concentration is roughly 4%, so it can live in very very salty conditions as well. So its example is a Helicobacterium, which includes several species found in salt lakes. Then we are having a thermophilic and sulfur reducing archaea. So they require hot water but differ in other habitat needs. So they thrive in only acidic water. That's why they also called thermoacidophils. And they require sulfur, calcium carbonate, and other live in alkaline springs. So they are thermoacidophile, they are heat and acid lover, and hyperthermophile, like they are having extreme heat lovers. So we are to, that was the first domain in which there are different categories of archaea, which are methogens, which are halophils, which are thermoacidophils also. Now we will talk about bacteria. So, first of all, we'll talk about bacterial ecology and diversity, where they are found. So, they live everywhere. They occur in water, fresh water, salty soil, air, and they live as saprophyte, parasites, and plants, animals, humans. They cause diseases, etc. They can survive in ice, hot water, salty water. They can form spores which are resistant to drought, chemical rays, and temperature variations. That's why they can live anywhere because they form spores on their body to protect them, and they can resist for longer period of time to drought, chemicals, and temperatures, etc. So some major groups of bacteria are, which we'll talk about in detail in its structure, that about gram positive and gram negative bacteria, and its major groups depends on its endospore formation, metabolism, growth, and nutritional characteristic, physiological characteristics, and some other criteria. But right now we are gonna talk about one most important category of bacteria, that is called cyanobacteria. Cyanobacteria, they are having the pigments chlorophyll A, phycobiliproteins, phycoerythrin, phycocyanin, blue-green color, which is present in phycocyanin pigments, yellow phycocyanin, and they store their food in the form of glycogen, that is present in its body, cyanophysin granules structure, and their cell wall is made up of amino acids and sugar. So they are commonly known as blue-green algae, and they are autotroph or like photosynthetic. That's why they are green in color. They found in the water area, damp places. Majority are free living also, but most are found in colonial. They are epiphytic, like they live on the plant's body. They are symbiotic nature also. So they contain a chlorophyll A, phycocyanin, that is blue, or phycoerythrin, that is red. They live in aquatic environment including oceans, pond, lakes, tidal flats, and moist soil. They live in colonies, filaments, and single cells as well. And the filaments forms can also move, and their example is Osculatoria, nostoc, anabina. So they produce gelatinous capsules which are often lighter than water, and they help keep the algae up near the surface of the water. So that can also be said to blue green algae because it is having blue and green pigments in that also, and there appears green, having blue pigment also, red pigments also. So it reproduces by fission, that is prokaryotic cell. They lack chlorophyll B, and photosynthetic product is stored in the form of starch, as we have seen before, that is similar to animal glycogen. If we see its structure, each cell is somewhat cylindrical or spherical. You can see its structure here, that is very clear, cylindrical or spherical, and in filaments form the cylindrical and spherical. They join together and to form um, long, that is called trichome. Trichome, this long filament is called a trichome, and trichome is having this type of cylindrical, spherical, and colorless empty cells. These are called heterocysts, and they are generally intercalary, like at some distances in young conditions, but they can may be terminals at the terminals here and here of this filament. Two polar two polar nodules are also present in each heterocyst. Polar nodules, we can see this is heterocysts. So these are polar nodules are present in these heterocysts. Some cells of the filament become enlarged, and they filled with food material, thick walled, and they are called the resting cells, and they are present in the chain form and filament form of this, and not that is cyanobacteria, and that is called echinids. Echinids is like a big circular or rounded cell may be present in uh, the center or at some distances in this filament or chain, that is called echinids, that is filled with a fluid material, and that can like bear lots of extreme conditions, because that undergo in rest condition as well as in the dormant condition for longer period of time. So that is very important for the survival of cyanobacteria. So this all filament is covered with mucilaginous sheath, which protects um, and just like protect this all filament from dehydration from external sphere conditions also. So if we talk about its importance, so that is very important in nitrogen fixation, because this is the symbiotic bacteria, so it is having their relationship with legumes, so it fixes the nitrogen very well. And then in Pakistan, that is grown on the large scale for the nitrogen fixation purpose. So first free oxygen organism we can say on the earth. So because that is blue green algae, so it produces oxygen, it does the photosynthesis like as the plants do. So they can be believed that they are responsible for first introducing oxygen into the primitive atmosphere. So they do the same process in non-cyclic phosphorylation in both photosystems. So they use carbon dioxide as a source of carbon, water as hydrogen donor for both of the photosystems, and they release oxygen using light energy. And they are said to be origin of the plants. Like this is endosymbiosis, like they are having the symbiotic relationship with the plants. So a photosynthesis, like they do photosynthesis, so they are said to be as primary producers in many ecosystems. So that is having great importance in for the ecosystem. So dear students, that's all for today. Inshallah, we will see you in the next lecture, in which we are gonna continue the topic